Hello, I'm Rick Roush, Dean of the College of Agricultural Sciences. We are excited to have you all here with us today for our College Connections webinar. These webinars are designed to give you a unique inside perspective of the diverse programs, people, priorities, and partnerships within the Penn State College of Agricultural Sciences. We are recording this session. You can find links to this and all past College Connection webinars, as well as registration information for future events on our college website by searching College Connections. Please note it does take a few days to edit and post the recordings after the session today. So today's session is from Breeding to Treat. Researchers discuss disease controls for tomatoes. I'm joined today by Dr. Majid Foolad, who's a professor of plant genetics, Dr. Kevin Hockett, who's assistant professor of microbial ecology, and Dr. Beth Gugino, who's a professor of vegetable pathology and the assistant dean for graduate education in the college. So after the presentation, we'll have a Q&A session, first addressing the questions that were submitted during registration. And we have a lot of them, particularly around diseases. If you have questions during the presentations, please enter them at the Q&A link, not the chat, because it's easier for us to track them in the Q&A. Now, before we dive in, I'd like to take a quick minute to introduce our first presenter. Dr. Majid Foolad has been a professor of plant genetics in the Department of Plant Sciences at Penn State since 1994. He conducts both basic and applied genetics and breeding research in tomatoes, including discovery and characterization of new genes for tomato response to biotic and abiotic stresses and for high fruit quality. Collaboratively with his students and others, he has developed the most detailed molecular genetic map of tomato and has identified and mapped genes for numerous traits, including resistance to early and late blight and improved fruit quality, such as high fruit lycopene content. Majid has developed many tomato breeding lines and hybrids and commercialized the tomato cultivar Valentine that won an All-American Selection Award in 2018. In the interest of full disclosure, I want to note that I personally had great success with the Valentine tomato in my own home garden, especially in a year of high blight problems. So with that, please take it away, Majid.
All right, thank you, Rick. As Dr. Roush said, I've been doing tomato breeding for a while. I've been at Penn State since 1994. So what I would like to do today, within a short period of time I have, just briefly describe what we have been doing, a little more about tomato breeding in general, and what we've been doing at Penn State. With that, I'm going to share my screen and go from there. So I hope everyone can see my screen, right? Fantastic. Fantastic. Thank you. Yeah, again, as Dr. Roush said, tomato actually is the most popular vegetable crops throughout the world, even if tomatoes were originated in warm climate, in particular South America, actually, Peru, Ecuador, and Chile, and it was domesticated actually in Mexico. Today, tomato being grown every corner of the world very widely, thanks to its adaptation, and also because of the different cultural techniques that we have to grow that. So tomatoes not only popular culturally, but also economically, they are so important. In terms of among the vegetable crops, often they rank #1 or #2, again, in our own states, in the US, in the world, #1 or #2 among all vegetable crops, and also among all crops normally rank 5th in the United States and rank 6th in the world. So economically, tomatoes are a huge crop. So with that, I have to admit that I've been working on tomatoes for a while. Sometimes it's not easy to grow tomatoes. It's easy to put them in the garden, but it's not easy to have a good crop or a good garden, because it's been reported that tomatoes are one of the most susceptible crops to diseases. There have been at least 200 diseases being reported that affect tomatoes, including all fungal, bacterial, viral, and nematode resistance. But fortunately, not all 200 diseases show up at the same time. From among these 200 diseases, we know about 10 of them which are the most economically, which are the most important diseases because they cause the greatest damage. Normally, worldwide, we spend over many billions of dollars to control the disease, as well as basically loss through the crop. Now, these 10 diseases, actually, basically 4 fungal diseases, 4 bacterial diseases, and 3 viral diseases are the most important one. In our own state, 3 of these 10, basically early blight, late blight, and bacterial canker, actually are the most important tomato diseases in our own state of Pennsylvania. And those are the diseases that I would be focusing on. Now, how do you control the disease? Basically, there are 2 major approaches to control the disease. One basically would be non-genetic, I call them non-genetic approaches, basically cultural practices, various cultural practices and chemical application, fungicide and bactericide or whatnot. Dr. Gugino will be focusing on those. My focus would be on using disease-resistant varieties. But I have to say, as a geneticist, as a breeder, I have to say that I won't expect that one day we have a tomato variety, or for that matter, any other crop that would not need a fungicide or would have complete resistance to all diseases and would not require fungicide. So by and large, the combination of these 2 approaches, means genetic approach in addition to basically cultural practices and application, in other words, an integrated approach, would be the most effective and efficient way of controlling these diseases. As I said, I'll be focusing only on breeding part of that, means using resistant varieties. But I just wanted to very, very briefly talk about how we, what are the different techniques that we use to breed tomatoes, and the same techniques actually are used to breed other crops as well. The first one is conventional breeding or traditional breeding. We've been doing traditional breeding for hundreds of years, at least 120 years since discovery of the science of genetics. And so still this is the most popular and the most commonly used approach today. The traditional breeding is basically based on phenotypic selection, means that you grow the plants and you evaluate them, you screen them, see which one is resistant, which one is susceptible. It takes forever. Normally, the life of a conventional plant breeding project is about 10 to 15 years. From the first time that you start doing your crosses to the time that you may have a variety to release to the market may take up to 10, 15 years. But then we have these other so-called biotechnology approaches, 3 types of those. The first one would be use of genetic markers. Genetic markers basically means that you basically fingerprint the plant. You take your plants using genetic markers, you fingerprint them, you know basically more about these plants through the markers than anything else. And so once you have these markers, and I'm going to show you actually a genetic map so that you know what I'm talking about. Once you have these markers, you do a process called marker-assisted selection. In other words, rather than phenotyping the plant, looking at the phenotype of the plant, you make your selection based on genotyping these markers, based on your fingerprint. This approach actually happened right now to be the most effective approach. I really don't know of any seed companies today, nationally, internationally, that they're not using market-assisted selection. But both of these techniques, conventional breeding and market-assisted selection, the transfer of genes actually are through sexual hybridization, means you make crosses and then you transfer the genes. But then beyond those, it comes transgenic and transformation approach, whereby you don't transfer genes through sexual hybridization, you transfer the genes through asexual means, through a vector, bacterial vector, viral vector, and whatnot. This technology, even if we don't use it much in my program, but I think is a very powerful technology, is a technology to be here. It's very unfortunate that we don't have very many tomato varieties today with transgenic approach. Not that the breeders are not interested, the breeders are very much interested. You could save millions of dollars by using this technology to develop new varieties, because once you have the resistant genes, you can transfer it to any genetic background that you want, to any variety that you want. Unfortunately, because of some resistance against this transgenic approach, we don't have, at least at the moment, we don't have. Recently, of course, you probably, many of you have heard that a new tomato was developed, a transgenic tomato was developed with high anthocyanin. This is basically purple colored tomato developed in England, and the US just recently allowed the use of that in the United States. That's a good sign. But my expectation is that, my projection is that within the next 10, 20 years, most of our tomato will be transgenic. So, and this transgenic approach could be very safe. Of course, you have rules and regulation can be very safe. But the latest technology that we use actually is so-called gene editing through CRISPR approach. What this technology is, unlike transgenic that you may transfer genes from one species to another one, in gene editing, you don't do that. You just, within your plant, within your variety, within your species, you just change or modify that genes, increase their expression, decrease their expression, or move them around within the species. So there is less regulations here. And so my expectation is that shortly, within the next few years, you will have a lot of tomato varieties, new tomato varieties that are developed to do gene editing. The only variety that I know today in the world actually that commercially available is one variety developed by Japanese breeder with particular amino acid, but I would expect within the next couple of years, you're going to see a lot of those in the United States as well. As I said, in our own state of Pennsylvania, there are three major diseases: early blight, late blight, and bacterial canker. And it happened that we've been working on all these diseases. And what I'm going to do actually take you very briefly through this thing, and in the Q&A, we can talk more about this thing. Early blight, early on when I joined Penn State in 1994, the first thing I did, I traveled around the state, talked to farmers and see what they were concerned about. Almost without exception, all farmers said that diseases were their main concern. At the time, early blight was the one, and that's why we established a breeding program on early blight resistance. They call them early blight because they normally used to show up before late blight, but right now sometimes we see late blight earlier than early blight. I tell you why that is the case. Normally, you don't see early blight early in the season. You don't see early blight before flowering or before fruit set, but right after fruit set, you're going to see early blight. Early blight is the disease that you're going to see every single year. There has not been any one single year in the past 28, 29 years I've been at Penn State. There has not been one year that I did not see early blight. So early blight has been and will be here. So we have invested significantly on early blight. Now, most varieties that don't have resistance, if you don't apply the forefront here, these are the varieties that were susceptible to early blight. You don't spray, you see the whole crop gets wiped out. But in the background, the main reason I'm showing you this slide, in the background, I have wild tomatoes. Wild tomatoes means tomatoes that have not been domesticated by breeders. These are basically widely grown in nature, and they have evolved with resistance. Much of my program has been getting into these wild type tomatoes, identify genes for various traits, including disease resistance. In this particular case, we did a lot of screening and the field condition, greenhouse condition, lab condition, and we have done a lot of genetic mapping. I promised you before that I'll show you genetic map. Like humans, we have 23 pairs of chromosomes. Tomatoes have 12 pairs of chromosomes. Chromosome number 1, number 2, and chromosome 12. To the right of each chromosomes are these genetic markers, the landmarks. To the left, actually, for this particular slide, are the positions of early blight resistant genes that we have identified. As you can see, we have identified several resistant genes for early blight resistance. Early blight resistance is one of the most complex disease resistance. Anyway, combination of these technologies used, we have developed both fresh market as well as processing tomatoes with early blight resistance. Within fresh market, we have developed different types of fresh market: the large tomatoes, plum, grapes, and cherry tomatoes. This is, for example, one shot of new hybrids relatively within the past 6-7 years that we have developed with early blight resistance. This is toward the end of the season, as you can see, loaded with fruit. And these plants, by the way, they're not sprayed with the fungicide. They show good amount of resistance. And then this is an example of our grape tomatoes. We've done a lot of work on grape tomato, as Dean Roush said. One of the variety that we have developed and commercialized in collaboration with the Johnny's Selected Seed was basically commercialized in 2018, and it won an All-American Selection Award. That meant that in 2018, Valentine was one of, if not the best of grape tomatoes, in the US or much of the US. So the hallmark of the main characteristics of this Valentine is that, first of all, we have high fruit quality. The hallmark of my program has been on fruit quality, developing tomatoes that are super red. So it has high lycopene content, but also it is early blight resistance is also pretty high in there as well. And I get a lot of good comments around the world. Actually, I've got good comments about this Valentine. So this is one of the varieties I would strongly recommend you to grow if you want. Then very quickly getting into late blight. Early on, when I joined Penn State for the first 10 years I was here, I had not seen basically late blight, but late blight, they call it late blight because it used to show up late in the season, early fall when temperatures cool down and you have high humidity. Late blight show up, but late blight is a lot more devastating than early blight. If late blights get into your garden and gets into your crop, not much you can do. Often, if you don't do any control measures, the late blight could wipe out your crop within 7 to 10 days, and this is a serious thing. It's not like early blight. It could wipe out your crop very quickly. So we've done a lot of evaluations. We screened thousands of actually accessions to identify sources of resistance. We did evaluation in the field, in the high tunnels, in the greenhouse, in the lab. This is one example of showing resistant varieties compared to resistant accession, actually the wild type. Resistance compared to susceptible. These plants were inoculated at the same time. As you can see to the left, susceptible lines were completely defoliated. To the right, these resistant lines actually were almost intact. So that was the start of a big project for us more than almost 15 years ago. And since then, we've done a lot of genetic studies, including identification of genes. There are basically 3 resistant genes, known major resistant genes for late blight resistant. They call them PH2, PH3, and PH5. PH5 we discovered at Penn State, but we have incorporated all 3 genes into our varieties. This is, for example, one of our breeding lines with only PH5 resistance comparing to a susceptible line without PH5. They were inoculated at the same time, and you can see the difference. But we have developed now hybrid varieties that have a combination of all 3 resistant genes, PH2, PH3, PH5. They are not in the market yet. I'm hoping that they will be in the market soon, but there are some other late blight resistant varieties on the market. I know that growers are very much interested in growing late blight resistant varieties. This is a list of the most common late blight resistant varieties that right now we have on the market. Both the plum shape, large size, small size, and different types of tomato. But there are more varieties, but these are the most common one. Now, very quickly on bacterial canker. Again, I had not seen bacterial canker in my field up until, 5-6 years ago. And now is one of the major diseases in Pennsylvania. So we jumped on it. We have done a lot of work on bacterial canker, and we have identified sources of resistance. And right now we're doing genetics and breeding studies. So with all said, I always get these questions that, hey, what's the best variety you recommend for growing in Pennsylvania? This is not an easy answer, because there is not one size fits all situations, because depending on whether you're interested in determinate varieties or indeterminate varieties, you're interested in open pollinated varieties, heirloom varieties, or hybrid. Early season, late season, you're interested in large tomatoes, small tomatoes, you're interested in high tunnels, greenhouse, or indoors, outdoors. So depend on the conditions, there would be different types of variety. But I have prepared this short list for you. These are the varieties that, based on the feedback from growers in Pennsylvania, they're happy with these varieties. Obviously, I strongly recommend Valentine, but also there are other, grape tomatoes. And the list of large fruited tomatoes, I've seen all of them basically under field condition. These are good, and we have some heirloom varieties as well, and then some plum tomatoes. But these are the most varieties that we've received, or companies received good feedback from companies. But I'm going to end up with this list. There are a number of other varieties that are being grown in Pennsylvania. I would be happy to share this list with you. My recommendation would be that if you're interested in and wanted to pick a variety, just go to the seed companies and look at the description of these varieties and see which one basically fits you, for indoor or outdoor, large size, small size, plum, grape, and whatnot. I'm going to end up with, finish my discussion with this last slide. In addition to diseases that affect tomatoes actually suffer from a lot of other non-disease issues, we call them, non-biological issues, because caused by environment, we call it physiological disorders. So these physiological disorders, and we can talk about them during the Q&A session, blossom end rot, which is very popular. I'm sure most of you have seen it, cracking, cat facing, and others. You can talk about these issues in tomatoes as well. My time is out, so I thank you for that, and I stop here, Rick Roush.
Thank you, Majid.
Thank you.
So, as I mentioned at the start of the presentation today, the talks are going to be recorded. So if you didn't get to write down all those names of the varieties you're interested in a couple of days' time, it'll be up and you can double check the list from online. So thank you very much, Majid. That's
thank you.
Tremendous amount of information in 20 minutes. Up next, we have Dr. Kevin Hockett, who's from the Department of Plant Pathology and Environmental Microbiology. His research focuses on the interactions between microorganisms living on the aerial plant surfaces, which is the phyllosphere, particularly the leaves, to understand the microbial ecology of the habitat and how to take advantage of these ecological interactions to help manage destructive plant diseases. So his recent work is focused on the development of disease-suppressive phyllosphere communities, which are conceptually like disease-suppressive soils that have been studied for nearly a century. He and his research group hope to develop an effective cocktail of microbes that can be applied to control the foliar diseases. Kevin has a bachelor's degree from Oregon State and earned his doctorate from UC Berkeley. He joined Penn State as a Lloyd Huck Early Career Professor in 2017. Over to you, Kevin.
Thank you, Dean Roush. Can you see the screen?
Yes.
Okay, great, thank you. Yeah, so as was mentioned, so my lab is particularly interested in sort of the microbial interactions and how we can take advantage of those interactions. And so today I'll be telling you about some of the work that I've been looking at, or my group's been looking at, to control a couple of common diseases that Majid mentioned in his last talk, bacterial speck and bacterial spot. So one thing to just start off and mention, when we think about disease, there's a number of factors that go into thinking about how much disease or the severity of the disease. And this is where, as plant pathologists, we rely on this idea of the disease triangle. And so this is just saying that the disease is really a product of three interacting factors. Do you have the pathogen present? That is, the organism that causes the disease. If so, is it aggressive? Is it abundant? And then there's the factor of the host. Is it susceptible or resistant? And certainly Majid just told us about a lot of his work trying to develop varieties that are more resistant to the disease. What I look at is more of the environment. And so I take the rest of the microbial community that a given pathogen has to interact with as being part of that environment. And so can we drive that in a way that makes it less likely for disease to emerge? And so one thing that we look at, again, there's a few different approaches to controlling plant diseases. And Majid told us about genetic host resistance and talked a little bit about, excuse me, cultural practices and chemical applications. But what my group is looking at is what we call biological control. So this is just using a live organism, typically in some formulation, applying it to the plant or maybe to the soil. And in using a number of different ways, that organism potentially would have the ability to exclude or inhibit the pathogen so that there's less disease. And so here's just a couple of examples. Here's Lifeguard. And so the active ingredient in this product is Bacillus mycoides isolate J. You'll see that this is OMRI listed, meaning that it's okay and can be used in organic production. That's one of the benefits of biological control, is that by and large, these approaches can be used in organic production. And so many people maybe go to any garden center and you'll see probably Serenade or something similar, this other product that I'm showing. And so this is another formulation, again, containing a live organism that is marketed to be used on a number of different plants, including tomatoes, and can be used both as a fungicide as well as a product that can control bacterial diseases, some bacterial diseases as well. So there's a lot of upside potentially to using biological control, but one of the major issues that's kept biological control from being more applied or more effective is just the year-to-year consistency. So some years there will be, you can apply biological control and it works very well, you get good control, but then the next season you may apply it and you get very little control. And so that's a major issue, right, for people producing a given crop or something like tomato. So what my group is thinking about is how do we really improve these biological control consistencies? So traditionally, most biological controls, we have a single organism. Those two products I just showed, they had a single organism in them that's really doing most of the work to control the spec pathogen. So that's the BCA, the traditional BCA approach on the top there. Now, one issue is that because you're working with a live organism, this organism needs to grow and be active in the environment. And if anything interrupts the activity of this one BCA agent, then it's not going to be able to do its job and inhibit the spec pathogen. So what my group is looking at is more of a whole community biological control approach. And the thought here being, if we have, say, 12 or 15 or a number of other different isolates or different strains that are all contributing toward control, then even if a couple of these strains maybe don't colonize as well, they're not as active when applied into the plant environment, that we have all of these other strains potentially available that can help still keep the pathogen controlled. And there's some idea for this from natural environments, as Dean Roush mentioned. We take a lot of inspiration from this idea of disease suppressive soils. And so what I'm showing here is for a given disease, this is a take all of wheat actually that we're looking at here. The disease severity for a given pathogen increases over time, but eventually, because it's consistently interacting with microbial community in the soil, eventually this disease severity comes back down, as long as the farmer is growing the same crop year after year. And eventually we get it to this disease suppressive soil where we have very low disease on the right side of this figure. That fluctuates from year to year, but it never spikes back up to a very high disease severity. Now, this has been studied again in soil systems for some time. And then the question is, can we translate what's known in soil environments to control foliar diseases? So things like bacterial speck spot and even bacterial canker as well, which can be a foliar disease. So what we've done is we've taken the approach of trying to develop these communities sort of artificially in the laboratory. We'll go out to a place in the environment and grab a natural microbial community from tomatoes. These could be tomatoes grown in someone's backyard garden or in a commercial setting or elsewhere. And we'll apply this community to a set of tomato plants and then introduce in the pathogen, either speck or spot. From this selection of plants, we look for those plants that have the least amount of disease. Then we collect the community from those plants and spray it onto the next round of new plants and repeat this process over and over again. And the hope is in the end, we get a community where there's less disease in this final passage and it's changed in some way that we can then replicate and utilize. And so this is actually what we've seen. So working with bacterial speck, this is work of a graduate student in the lab who's recently graduated. But what we're showing here is the disease severity for speck going up over these passages until it reaches about passage 4, and then it comes back down and stays relatively stable until we got to the end of the experiment. We've also done this with bacterial spot as well. So here we're looking at comparing. So this is a healthy plant, didn't have any pathogen inoculated, the control on the left. The community here, this plant had a suppressive community applied to it before the pathogen, and there's still clear disease coming through. But if we compare this plant to the one right next to it where there was no community added, we can see that this plant is actually in a healthier condition. Again, still disease, not perfect, but it's doing better than if that community hadn't been there. So this is what we've done over about the past couple of years. And so now what we're looking to do is we think that we have good evidence that we can develop these disease suppressive communities again that'll be natural. Well, they're developed communities, but they come from natural sources. There's no genetic manipulation or anything like that going on. So any product coming out of this theoretically would be could be applied in organic production settings. But the real question is, okay, now that we have these communities, can we really apply them to an agricultural setting? And can we, I talked about bacterial speck and spot, but can we also develop them against something like bacterial canker, which was, Majid mentioned is a major issue in the state. And then even thinking about maybe some other foliar diseases as well, some of the fungal diseases potentially. And so that's really where we're going with this work. So I'll just end and highlight the people that have really worked on this. So Dr. Hanare Ihotomonu recently graduated, and she really started this work off. Constanza Bartolomeo Koninks is a student who just finished with her master's, and she did the work in bacterial spot. And Dr. Beth Gugino has collaborated on this work and was the co-advisor for Constanza. And this is the sources of all of the funding, and I just really want to highlight the Pennsylvania Vegetable Marketing and Research Program. They funded this work very early on and have been very helpful in getting this work off of the ground and getting us to a place where we think we have something to really try at the next level. So with that, I'll stop. Thank you.
Thank you, Kevin. That's terrific. Quite a eclectic group of funders there for that project. That's wonderful as well. So last but not least, we have Dr. Beth Gugino, who's a professor and vegetable crop extension plant pathologist in the Department of Plant Pathology and Environmental Microbiology. Since 2008, her extension and research program has focused on understanding pathogen biology, epidemiology, and developing and translating integrated management tools for important and emerging diseases of vegetable crops, including tomatoes. And we're thrilled that as of last fall, she's also taken on the role of Assistant Dean for Graduate Education in the college. Thank you, and off to you, Beth.
All right, thank you. Go ahead and share my screen. Okay. Yeah, so today I'd like to share a little bit about our applied research program for tomato disease management. And as Majid and Kevin have shared, there are a lot of common diseases of tomato. I'm also going to throw into the ring septoria leaf spot, as well as anthracnose, which is a fruit rot. Also, there are a number of common disorders of tomato. We talk a lot about blossom end rot. We talk about physiological leaf curl, which when we think about in June this year, it was really incredibly hot early on, and we had a lot of this severe leaf curling. We also have walnut wilt. So if you're growing tomatoes in a garden and you're near a walnut tree, walnuts and tomatoes don't mix at all, and it can take your plant out really quickly. But all these pictures are to share the importance of diagnostics and understanding, what problems you're having with your plants in order to identify, what's going to be a successful management solution. And so we have the Plant Disease Clinic here at Penn State. Dr. Sarah May and Jenny Mazzoni both work there. It's a free service for anyone in the Commonwealth to submit samples for diagnostics. We also have an excellent Master Gardener hotline that you can always reach out and ask them questions, and they can help out quite a bit in terms of diagnosing what's going on, especially with tomatoes, since they're so common, in home garden settings in particular. I'd also share that it's important to kind of stay informed with what's going on across the state. I work very closely with commercial tomato growers, and so we have provided a number of different resources so that they can stay on top of when are we starting to see early blight? Are we having outbreaks of late blight? We haven't seen late blight for a number of years now, but that's always one that everyone's very concerned of for the reasons that Majid shared. We have a 1-800-PEN IPM hotline that you can call and get leave an auctioneer message about what we're seeing in terms of tomato and potato production. We have a podcast series, as well as various newsletters and information that we post at the produce auctions, because we have a lot of Amish Mennonite producers across the state. So trying to get information out there so that growers can make, decisions, in-season decisions in terms of disease management. And like Kevin shared, the disease triangle is really the foundation of everything with disease management. And I think I've included this slide in every single talk I've ever given in my entire life. But really, when we're thinking about managing, we're thinking about eliminating the pathogen from the system. We're thinking about altering the environment, and also thinking about host susceptibility. So if we break up this triangle in one or multiple places, we're going to be able to reduce and manage the disease. And so this is kind of where my program falls in. So when we think about disease management, we also think about basically the IPM triangle, where the majority of the tactics that we have for disease management kind of fall at the base of this triangle. So a lot of what we're trying to do is prevent disease from developing in the first place. And so, Majid's work with developing resistant varieties is really critical here. This is really the foundation for disease management, is being able to select resistant varieties. We also think about grafting. So for soil-borne pathogens, we can graft onto rootstocks that can help then those plants overcome those disease issues. We also have practices like crop rotation. I know there's a lot of questions in a small garden situation. How do you actually rotate a crop? In those small situations, it's really important to think about sanitation. So really trying to remove as much of that crop residue from the garden as possible. If you're going to be continually planting tomatoes in a very small area. Also thinking about things like drip irrigation. So the worst thing you can do is have those plants, the leaves of the plant be wet for an extended period of time. So really the drier the leaves are, the longer, the better, the less disease issues you're going to have. So things like drip irrigation, things like using mulches to prevent rain splash. These are all tools that can help prevent disease to begin with. And then once we've identified disease and we, are seeing it in the field or even preventatively, we might move up into these biological products. So things that Kevin was talking about in terms of developing, microbial consortia that we can use to help reduce disease pressure. And then we move up to the top of the triangle, which is using conventional chemistries. In my program, we will work with both using biorational as well as conventional chemistries to try to reduce disease pressure. And so one of the tools that we've been working with, and this is thinking all the way back to the seed, because we know that pathogens can be associated with the seed, is the idea of using hot water seed treatment to help manage bacterial pathogens. So basically the idea is you're taking that seed and you're submerging it in water at a certain temperature for a certain duration, and you're basically taking the seed kind of to the brink of its life, and then you're bringing it back. And in the process of doing that, you're able to kind of rid that seed of some of those bacterial pathogens. In this picture here, this is bacterial spot on tomato transplants. These plants, you may think you can kind of limp them through the season, but this is really a never ending battle. They'll never win the battle with the transplants that have bacterial spots. So this is one tool that we're trying to use to educate growers to help reduce some of the bacterial disease pressure that they're seeing. Another strategy that we're using is part of an IPM approach, and this is really working with soil-borne pathogens. We grow a lot of tomatoes in Pennsylvania in what we call high tunnels, and so that's what this structure is. It's not quite as sophisticated as a greenhouse. You can roll the sides up and down. You can have end walls, allows for rain protection, but also allows for water or for air movement. One of the challenges is growers like to plant tomato after tomato after tomato after tomato after tomato in these types of settings. And so then you end up with a buildup of soil-borne pathogens in those cases. And so this process of anaerobic soil disinfestation, the idea is you're adding carbon sources to the soil. You're mixing them in. So in this case, we're adding poultry litter, we're adding liquid molasses, we're tilling it in, and then we're covering it with this impermeable film and flooding the soil. And that creates anaerobic conditions that basically knock back the pathogen population. So this is something that we're looking at in high tunnels. This work is with Dr. Francesco De Goya in plant science, and my graduate student Katerina. And I'll point out too here that the Vegetable Grower Association and Vegetable Marketing and Research Program has really been important in kind of helping to get some of this funding going, this project going. The other kind of aspect that we focus on with tomatoes is doing product efficacy trials. So there's a lot of products out there on the market that growers can use to manage these various diseases. We look at evaluating experimental products that are in development, newer products that are on the market, how effective are they, looking at programs that use reduced risk kinds of products, as well as looking to monitor for fungicide resistance. So really this is helping to provide unbiased data to share with growers so that they can then make informed management decisions. And this is just an example of one of these trials. This is looking at early blight and Septoria leaf spot on tomato. In the bars here that are green, we're getting a very high level of management, reduction of disease. So we're able to, in here, this case, the top one, this experimental biological. So it's a biological control with a couple of our typical conventional products. We're able to get, 97% control of the disease. And so we're able to then use, this information and then, well, this is just some pictures of what it looks like when you don't manage the disease. So in this case is an untreated control. So if you don't do anything to try to manage early blight and Septoria, you can obviously have a significant amount of defoliation. In this case, these fruit are never gonna reach maturity versus our experimental biological with Quadris or our kind of our grower standard. So in a lot of cases, when we don't use host resistance, we're often really reliable on these types of chemistries. But then all this information feeds into our Mid-Atlantic Commercial Vegetable Production Guide, which is a regional joint publication that provides information for our growers in terms of helping them select products for managing these various diseases. And so I just want to kind of end here with, from a homeowner's standpoint, because I know a lot of you are homeowners with vegetable gardens growing tomatoes. There are, a number of products that can be used to help manage these diseases in a garden situation, in addition to some of the biologicals that Kevin mentioned in his talk. But with all of these different products, it's always important to keep in mind, and this is where the extension plant pathologist comes in, that these all have labels to them. So it's important to be, following that label and making sure that you're applying that product correctly and according to label recommendations. So it helps keep you safe and it helps to manage those various diseases. So, and with that, I'll just say, have a happy, healthy, and successful tomato growing season.
Oh, thank you very much, Beth. So now, and really foreshadowing that we have a lot of questions about growing and controlling diseases in tomatoes. So it's time for Q&A. There were about 50 submitted. Might just run through a couple of them quickly, with very short answers. So one of the questions was, what organic soil conditions and organic fertilizers are most beneficial for tomatoes, and what fertilizer is best? And it's really about soil testing, isn't it? You need to do soil tests, find out what you need in just that amount, right, Beth?
Yep, I would agree with that. And I would also say, with compost, more is not necessarily better. Compost is a great source of organic matter, but if you put too much on, you can create conditions that are actually very unfavorable for the plants.
Thanks. Another couple of questions, Majid, should you thin tomato plants, or how should you prune a bush type tomato compared to a regular vine tomato?
Yeah, thank you. Definitely you want to prune your tomatoes. Assume that they meant pruning rather than thinning. Thinning is different, but definitely you want to prune your tomatoes. And the way that you prune your terminate tomatoes is different from indeterminate. The determinate tomatoes, by and large, they don't need a lot of pruning, except that you want to basically cut and remove all the lower leaves. But technically, you want to all the leaves and branches below the first flower branch or the first cluster. I would remove all of those. Then don't do anything else. So that's easier for determinate. For indeterminate, again, as the name indicates, if you don't prune them, they grow forever. They produce so many different lateral branches. Pretty soon you're gonna end up with the jungle of foliage. You don't want that. So my suggestion that you do want to have only one or two so-called leader stem or leader branches, 1, 2, maximum 3 normally. So what you want to do, first of all, again, the same thing as determine all the lower leaves below the first, first 5, 6 inches. I would remove all of those for sure. The main thing is that you want to allow air flow in the plants. As Dr. Gugino said, so cultural practices actually is very important to prevent diseases. One of the most important cultural practices is to allow air flow inside the plants. If you allow all these branches, you're not going to have too much foliage, you're going to have a lot of diseases. So with the indeterminate, I would leave just 1 or 2 leader branch and all so-called the suckers. The suckers are these side branches that are at the armpit, between the leaf and the main stem. I would remove all of those early on and would only keep 2 main branches. And then you can stake them, depend on 1 branch, 1 main stem, 1 leader stem, or 2 stem. You need 1 stake or 2 stakes, and you go from there.
Yeah, thank you. My cherry tomatoes sound like the ones you described that turn into a jungle if I don't prune them. So
yeah, if you don't prune to do that, it happens, right?
Yeah. We've got a lot of people who will be growing tomatoes in small space, and they're not really going to be able to rotate really well to avoid disease buildup. What can they do in order to try to reduce their disease problems?
Yeah, this is where I think sanitation is really important. So making sure that, at the end of the season, you're really cleaning all of the crop residue out of the garden. So a lot of the foliar pathogens we were talking about, except for late blight, will survive with the crop residue. And so that's why it kind of will keep coming back year after year until that plant material thoroughly decomposes. So really, sanitation, and then also making sure you're selecting varieties, that have resistance to them. That will help a lot as well.
What varieties tend to produce a large quantity of tomatoes, not just a few per plant, Majid?
Yeah, I would say, the hybrid varieties, modern varieties, normally produce a lot more fruit than heirloom varieties. The heirloom varieties, the plants tend to be larger, but the number of fruit tend to be, smaller in number. Sometimes I joke with my students in the field, you say that in hybrid varieties, you may get up to 50, 60, 70 fruit, but or at least 30, 40 fruit, but in heirloom varieties, you may get 3 or 4. They may taste better, flavor better, but you're gonna have less fruit. So depend on what your hope or what your goals are. If your goal is to have more fruit, I would grow hybrid varieties. If your goal is to have more taste and the number of fruit is not important, I would grow heirloom varieties.
Good. So does it matter? Are there some tomato varieties that are best for sunrooms and so forth, or does it really matter?
Oh, no, it does matter. There are some varieties that do better, in indoor compared to, outdoor. But, a lot of varieties that normally we develop for outdoor, people also would grow them indoor as well. But there are some varieties that are grown indoors. In particular, if you have indoor, normally people grow more indeterminate varieties because they can tie them up, they can stake them and everything. Outdoor, normally they grow determinate plants.
So tomato seeds, is there a lifespan you can have for tomato seeds? And if you're harvesting your own, how you go about it?
No, that's a good question. Actually, tomato seeds under room temperature, if you clean them first, after you collect the seed and harvested the seed, if you clean them well and bleach them, normally at room temperature you can keep them at least a couple of years. But if you keep them in so-called, the seed storage facility, this is about, the temperature wise about 40, right about freezing point, about 40, and then humidity about 40, less than 40 or 40, 45. You can keep them for 10 years at least, if not more. The seed viability gradually goes down. There is no doubt about that. But you could keep your tomatoes in seed storage facility at least 10 years. You can keep them for long term, in frozen conditions for hundreds and thousands of years, hundreds of years. But no matter where you keep them, viability normally goes down. some growers, some farmers actually keep their seed in the refrigerator. That's okay in terms of temperature wise. It's okay, but in the refrigerator you normally get high humidity. I would put them in a jar, maybe add some desiccant to them. If you keep them in your refrigerator with some desiccant, I think you can keep them at least 10 years.
That's great. One of our questioners really sympathizes with the life of a breeder. What's the length of time, I think they mean in years, to produce a new variety of tomato or potato, both of which in the same family?
Yeah. So, tomato actually, it's long stories and a lot of interesting stories about tomatoes being from the same families, because even if tomatoes were introduced in North America from Europe, actually, first of all, let me back up. So they moved from South America first to Europe, and then from Europe to the United States. And because it was from the same family for a long time, people thought that it was poisonous and they were not eating them. Actually, it's not more than 100 years since we started actually eating tomatoes, in the United States. So, but in terms of the tomato fruits actually don't have much, if any, poisons, particularly ripe fruit. You may have some, poisons, in the tomato, they call it, in the foliage, but definitely not in the fruit. So the fruits are pretty much safe, even if it is from the nightshade families. So yeah.
Average new potato variety, a dozen years at least?
Yeah. Oh, yeah. How long it takes? It takes actually, again, as I said, it normally takes between 10 to 15 years on average to develop new varieties. In case of potatoes, sometimes you clone potatoes, so that might be easier. But if you want to develop new varieties, hybrid varieties and all these things, it may take as long or longer as well. Right. But conventional breeding, about on average 10 to 15 years. The market assisted selection, actually, you could cut down half of that, 5, 6, 7 years. The transgenic approach, if you have the gene clone, could be much faster as well.
Right. So Beth, another question for you. How do you identify early and late blight? Or maybe another way of looking at it, if the treatment tactics are the same, does it really matter that much if you can identify one from the other?
That's a good question. So, early blight, I think like Majid said, usually you'll see early blight with the onset of fruiting. So when the tomato plants start producing fruit, it'll start with the lower leaves and will tend to defoliate the plant, working its way up. Late blight, and it produces more yellowing on the leaves, and the lesions are more like little targets with concentric rings to them. Late blight, like I said, is, everyone says I have the blight, right? And it's like, okay, which blight are you talking about? Late blight can really affect the plant at any growth stage, and it tends to affect maybe the more interior or center part of the plant where there's more moisture. The leaves are wetter, longer, and the lesions, it's much more of a greasy gray, brown color. They look very, very different. So if you're in the management, yeah, you're right. The management would probably be the same, but I would say that we don't really see late blight that often right now. And if we did see it, that's one of those things where all of the alerts and information that we try to get out through our various networks, we would try to alert people pretty quickly about late blight.
So does the blight ever go away in garden soil, or does it just gradually diminish over time?
So again, it depends on the blight. So late blight is an obligate pathogen. So it requires living plant tissue to survive. So in the case of tomatoes, once that tomato plant is dead, the pathogen is dead. In the case of potato, late blight could survive associated with that potato tuber, and that's one way that it can move around. Early blight actually survives with the crop residue, and so it's allowing that crop residue to thoroughly decompose. And once it's thoroughly decomposed, the pathogen really can't survive in the soil on its own for very long.
That's good news. Okay, so why do tomatoes get black on the bottom and rot? What's causing that?
So a lot of times that's what we call blossom end rot, and it's not a disease. It's a calcium deficiency at that bottom part of the fruit, and so calcium is needed for cell wall integrity. And a lot of times it's not again, it's not a calcium deficiency in the plant per se or in the soil per se. It's the plant having difficulty getting the calcium out to those outside fruit. And so it's more often a watering issue, so irregular watering, or it's too much nitrogen and the plant is growing incredibly vigorously and can't get the calcium out to those end fruit.
So yet again, use the right amount of fertilizer.
Yeah, one other thing I could add, exactly what Beth said, I would normally we use also so-called CalMag fertilizers. So fertilizers with additional calcium and magnesium, so that calcium actually prevents a lot of that blossom end rot.
Great. And you've identified varieties that are reasonably resistant to early and late blight. That's in your list on your PowerPoint. Are there any when you mentioned there's one genetically altered tomato that the Japanese are using? I think in the past people have made virus-resistant tomatoes, but they never really were commercialized, right?
Exactly. No, the University of Florida and then some companies in collaboration with the University of Florida, they have developed nice varieties that have not been commercialized with resistance to a number of viral diseases, in particular TYLVC, tomato yellow leaf care virus, which is devastating tomato crop in Florida. But again, unfortunately, no grower is willing to take these varieties and grow these varieties. So, but yes, there are experimental transgenic tomatoes with disease resistance, in particular viral resistance, a number of other things. But commercially, we don't have them yet.
Yeah, and they've been around. Maybe Mill Zeitlin was creating some of these back in the late 80s, right? So
I remember I was a graduate student in early 1990s with Flavor Saver, Calgene company at UC Davis, and in Davis, actually, the company at the time was, they developed this Flavor Saver, had a longer shelf life, work on this gene polygalactoronase, so it increased the shelf life of the tomato. But the unfortunate thing at the time was that these people who were expert in transgenic, they were not expert in plant breeding, so they put the gene in the wrong background. So the tomato was no good. It has good shelf life, but the tomato was not good. So that was a big failure, and that's what a very big disappointment for transgenic. Actually, that was the first transgenic plant. So that was very unfortunate.
So is it true that using salicylic acid or powdered milk or these or other kind of home remedies, will they do any good on these, Beth?
So from a home remedy standpoint, I probably would say I'm less knowledgeable about that, since I work primarily in the commercial grower realm. But what I would say for a home gardener who's wondering what may or may not work is that maybe you could try something on one of your tomatoes and not on one of the other ones, and see if you're able to see less disease, maybe less early blight on the plant that you treat versus the one that you don't treat. I know there's a lot of information out there on the internet with home remedies for plant diseases.
Yeah,
right.
And
well,
I could add somewhat to that in my earlier life, when I was working on abiotic stresses, salt tolerance, and cold and drought tolerance, we wrote a review paper on the effect of growth regulators on improving plant stress tolerance. Our focus was on, at the time, was salt tolerance, but one of the growth regulators we wrote about was salicylic acid. Through our journey studying salicylic acid, we learned that salicylic acid basically improve the whole stress tolerance in plant, both biotic as well as abiotic. So I agree with Dr. Gugino that yes, so that's basically adding that. Like some people just use aspirin. Sometimes they spray plants with the aspirin, and they see some improvement, in this, stress tolerance. That's scientifically not been proven, but some of the growers, claim that that's the case. But that's basically what the effect of salicylic acid is.
that's right. Okay, so we've run out of time. I first want to thank Majid, Kevin, and Beth for their outstanding presentations today and great answers to questions. Up next for College Connection in July, we'll be hosting Seth Bordenstein, who's the director of the Microbiome Center, and Sarah Bordenstein is associate research professor, to discuss the Penn State Microbiome Center and its research, along with their flagship science education project called discover the microbes within. So this is a great husband and wife team in the exciting area of microbiomes. And you can check back at the College Connections website for registration information. So we look forward to seeing you again soon, and thank you for joining us. Have a great afternoon, and yet again, thanks to Majid, Kevin, and Beth. Thank you.
Thank
all.
you.
Have a
Thank
great
you.
afternoon, everyone.
Thank you.